Hi guys, in this video you will learn about citric acid cycle. But before going to citric acid cycle, I'd like to bring your attention to the structure of mitochondria, which is where our citric acid cycle and the electron transport chain in the next video take place. So mitochondria, like chloroplast, is once a free living organism as a prokaryote. But at some point, they got eaten by a eukaryotic cells and become part of it. They live together. The mitochondria provide energy to the host, and the host provide food to mitochondria. But that's all about history. So let's get into the structure. Mitochondria compose of two membrane system. The outer membrane, like the outer capsule, and the inner membrane, which is more like very wavy. And you have three spaces. In the innermost is matrix. Somewhere in between is intermembrane space, and the other one is just called intercrystal space, which is like a pouch that combine the intermembrane space and enclose it with the inner membrane. All right. So in most of the part, citric acid cycle occur in the matrix or in the most space, but the in the electron transport chain, electron transport chain occur in the inner membrane, and that's also involved in creating the proton gradient between matrix and intermembrane space. All right, so let's get started with the citric acid cycle. From glycolysis, you get either pyruvate or malate. If you get malate, it need to take off one carbon molecules to become carbon dioxide, and it will release one electron to produce NADH, and it will become pyruvate. This pyruvate can also lose one carbon to become carbon dioxide and lose one electron and produce NADH, and it will become carbon 2 molecules called acetyl-CoA. This acetyl-CoA can combine with another product from malate. Malate can also lose one electron to become just NADH, and it will stay C4 again and become oxaloacetate. Oxaloacetate together with acetyl-CoA, 4 carbon plus 2 carbon, and adding one more water molecules, it will become carbon 6 compound. It's called citrate. This citrate can be losing carbon dioxide twice. Firstly, it will be converted into isocitrate, just an isomer of citrate, and then losing one electron, losing one carbon, and become alpha ketoglutarate. And you can notice that it is composing of only 5 carbon right now. Again, it losing one electron and one carbon as a carbon dioxide and becomes C4 compound, which is succinyl-CoA. Later, succinyl-CoA can losing one CoA. CoA is just a coenzyme. It's called, the full name is coenzyme A. By losing one coenzyme A, it produces ATP and becomes succinyl-CoA. You can notice that from this point on, it will not lose carbon dioxide anymore. Succinate will produce fumarate and also lose 2 electrons and become FADH2. Fumarate can combine with water and become malate to complete the cycle. In this cycle, I like you guys to notice the direction of the arrows. Some arrow has just one direction. This means the process goes just one way. If the process contains two direction arrows, this means the direction of the reaction can go either forward or reverse. Like this one, it can go both ways. And again, this process making ATP is called the substrate level phosphorylation. Let's take a look at the enzymes. First, pyruvate, losing one carbon and also two electrons to become an ADH, to produce an ADH, and become acetyl-CoA with an enzyme pyruvate dehydrogenase. Combining of acetyl-CoA with oxaloacetate to produce citrate. You need an enzyme citrate synthesis means synthesizing citrate. From citrate to isocitrate, you need an isomerizing enzyme is called aconitase. Isocitrate to alpha-ketoglutarate, you lose two electrons and it will use isocitrate dehydrogenase. Losing another two electrons with alpha-ketoglutarate dehydrogenase from these two enzymes. The name of the enzyme is actually came from the substrate, either isocitrate for this enzyme or alpha-ketoglutarate from this enzyme. And the type of the reaction is redox reaction or dehydrogenation. So the enzyme named after dehydrogenase. And then succinyl-CoA to become succinate. This process is convertible, and the enzyme's name is succinyl-CoA synthetase. They are similar, the name is similar to synthase, but synthetase is just mean producing something but require ATP as an energy. As I mentioned, this process is reversible. So if you want to make succinyl-CoA, you need an enzyme. You need ATP as an energy, and the name of the enzyme is synthetase. This means you need energy. Without energy, the name of the enzyme will be just synthase. With an energy, the name of the enzyme is containing the in the middle become synthetase. Alright? Succinate to fumarate, you need dehydrogenase. Fumarate to malate is just adding one water molecules. You use fumarase. Malate to become oxaloacetate is dehydrogenase. Malate to become pyruvate. This one is an exception. You lose two electrons and also carbon. And this enzyme is NAD plus malic enzymes. I cannot find explanation for this name. But anyway, this is the end of citric acid cycle, and in the next video, it's gonna be longer. I promise. It will be about electron transport chain in plants.